Hello everyone, it's Dr. Rafael Gutierrez. Today I'm going to talk about the basic carbon chemistry, which is called the organic chemistry. And what I would tell you is on the periodic table, anytime you have it, anytime you're doing chemistry, keep one in front of you. And remember, you have a +1 here, a +2 here. On the carbon, you have a plus or -4. On the nitrogen, you have a -3. On the oxygen minus column, you have a -2. And on the chlorine or fluorine, you have a -1, and then you have the noble gases that don't have charges. Now, before we actually talk about the carbon chemistry, the organic chemistry, there are things that are important to know, and that is that when we are mixing things together, a lot of times we want to know how many atoms are in a thing, are in a mixture. And so we actually look at the moles. Now, moles is a measure of atoms based on Avogadro's constant number. Pretty much a mole is equal to 6.022 times 10 to the 23rd atoms. It's a huge number. And so usually we end up using that. The reason we get that number is because what we want to do is we want to get something that tells us how much something would weigh as we measure it. And so what ends up happening is if we use the molar mass of something and just say instead of molar mass, we say the molar instead of saying the molecular mass, we say the molar mass, I'm sorry, we actually are saying how many kilograms per mole. And so if we have 12 kilograms of carbon, for instance, if you look back at your periodic table, you'll see carbon is 12. So 12 kilograms of carbon would be equal to 1 molar mass of carbon, 1 mole of carbon. If we look at different things, we actually sometimes have to bind them together. So for instance, if we're looking at hydrogen gas is composed of 2 hydrogens. And so the molar mass of hydrogen gas would be 2 kilograms per mole. We also have molality, which is the amount of moles that we actually dissolve per liter. So if we have a molar mass in grams, we can calculate what its molality would be. Now, when we're mixing mixtures like that, usually we have something that is being dissolved and something that dissolves. Together we call this a solution. A mixture of things that are dissolved with what dissolves it is a solution. And in a solution, the thing that's being dissolved is called a solute. And the liquid that's dissolving the solute is a solvent. And so always look at that. Now, one of the big things that we can look at is ocean water. If we think of ocean water as mainly salt water, it has sodium chloride as our solvent and water is our solvent. And so we have a solution which is sodium chloride and water, with the solvent being water and the solute being sodium chloride. Another thing that's actually important to look at is diffusion. Now, diffusion is a vital concept to understand life and how everything in life works. The best way to actually think of it is molecules vibrate. And because they vibrate, they tend to move from an area of high concentration to low. Best way I can describe this is think of your favorite food. If you, let's say, want pizza. If you think of a pizza on a table and you're far away from it, you might smell the pizza a little bit. And as you walk closer, the smell of pizza gets stronger and stronger. The reason is the particles of pizza are diffusing through the air from a high concentration where the pizza is to low everywhere else. And it ends up forming a gradient, meaning that there's going to be areas with higher concentrations and low based on how close it is to the highest concentration. Another way to look at this is by taking a teaspoon of water, taking water and putting a teaspoon of salt in it. By doing that, if you drink it, you'll see that the water becomes salty. Now remember, this is a passive process, but if more heat is introduced, diffusion will increase. The other thing that's important to understand when we're dealing with organic chemistry, actually any chemistry, is the difference between acid, base, pH, and buffer. Now, an acid is something that gives you a hydrogen ion, so it gives you an H+. A base is something that accepts a hydrogen ion, so it removes an H+. And neutral is when the acids and bases are even. And so if you look at water, water is neutral. By the way, it can give you a hydrogen ion and it can accept hydrogen ions. And so that's actually neutral. And when we're measuring the pH, when we're measuring the acidity of something, we use the pH, which pH stands for potential hydrogen. It's actually looking at how many hydrogens are potentially in a solution. If we have more base, the pH goes down. I'm sorry, if we have more base, the pH goes up. If we have less hydrogens, the pH goes down. Now, something neutral will be water because of the mathematical equation that's used to calculate pH. But for now, just remember pH is a measure of hydrogens. If you have more hydrogens, you have lower pH. So pH of 7 is neutral. So if it's less than 7, it has more hydrogen, which means it has a higher pH. If it's more than 7, it has a higher pH, which means it has a lower amount of hydrogens. Now, there are a couple examples of acids that I like to talk about, and one of them is carbonic acid. We get carbonic acid by mixing carbon with water, and it will bind and give you H2CO3, which you can see has enough carbon and hydrogen and oxygen to give you carbon dioxide. When this is in water, the hydrogen can come off, and so it's an acid, and so you end up with the hydrogen and what's called a bicarbonate. Another one people like to talk about is hydrochloric acid. You have a hydrogen and chloride, and when this is put in water, the hydrogen comes off, the chloride comes off, and so you have a free hydrogen ion. There are bases, and I'm going to actually use one of the same bases that I already talked about. Over here you can see the carbonic acid. So if you have bicarbonate and you have hydrogen ions, the hydrogen ions will bind to the bicarbonate, giving you carbonic acid. We also can talk about sodium hydroxide. Hydroxide tells you 1 hydrogen per oxygen, and so you can see it here. Sodium here. And so when this is put in water, when this is put with an acid, the acid will bind to the hydroxide, giving you water, and the sodium will come off. So you can see how it's accepting hydrogens on this side here. Now, a buffer is actually kind of interesting because it's a solution that has both a hydrogen acceptor and a hydrogen donor. And usually the hydrogen acceptor and donors will work together to maintain a relatively constant pH. Over here you can see carbonic acid and bicarbonate. What can happen is this hydrogen ion can be donated and bind to this bicarbonate, making it carbonic acid, and the carbonic acid that donated the hydrogen would be a bicarbonate. And if an acid were added to this, the bicarbonates would actually neutralize the hydrogen. If a base was added to it, the hydrogens would neutralize the base, and so the pH maintains relatively constant until the system is overloaded. If we look at the body, over here you have the C6H12O6, which is basic sugar, and unlimited oxygen, and you can break it up into carbon dioxide and water, which are going to bind, giving you carbonic acid, which you then have bicarbonate and hydrogen. In your body, the hydrogen usually will bind to some sort of protein, and so what you end up with is you have bicarbonate and carbonic acid, and so your blood will actually have a pH buffer within it. Now, I want to start now talking about what are called macromolecules. Macro means big, so these are big molecules, and we use these for a lot of things. The first ones I'm going to tell you are going to be the hydrocarbons. Based on the name, you can see hydro means hydrogen, carbon means carbon, obviously. And so a hydrocarbon is something made up of hydrogens and carbons. And what ends up happening is this: as carbon can potentially bind 4 times, you will end up having carbons bound with hydrogens. I'm not going to draw the hydrogens, I'm just going to put this here. And sometimes this bond here will bind to another bound here, and so you can end up having a double bond. Now, sometimes those double bonds are not on the same side. And when you take organic chemistry proper, you will talk about how there are different configurations of this and how you can end up making ring structures in carbon. And you can put one hydrogen up here sometimes, and if this is a ring configuration, meaning they can't rotate, if you put a hydrogen ion over here and the double bond is here, so we'd actually have to get rid of this line here, we would end up having the hydrogens on opposite sides. As they're going through the carbon structure, we call them a trans configuration. Trans means it goes through the structure. A cis would be if the hydrogen, instead of being down here, would be up here. And so the hydrogens, let's put an H here. The hydrogens would be on the same side, so that would be a cis configuration. Hydrocarbons we tend to use as fuels, and the really most elementary one is methane, which is one carbon with 4 hydrogens, which can actually form spontaneously. Now, here I actually tell you hydrocarbons we use for energy. The most elementary is the methane, which is composed of carbon and 4 hydrogens. As more carbons are formed, we actually do have more variations. And one of the neat things about this is these tend to be nonpolar. What it means is they don't have a negative side and a positive side. They tend to be more neutral. And so most of these hydrocarbons are not soluble in water. They're hydrophilic. And so what happens is if you have a nonpolar molecule, it will be hydrophilic. Now there are some things that are different. Again, I told you about cis and trans. I would tell you on a separate piece of paper to define these two, both as you would actually explain it and then what it means in chemistry. Methane is, like I said, the most elementary, and it can form spontaneous under low pressure with carbonic acid or through anaerobic respiration in some bacteria. Now the interesting thing about it is if you have more methane in the atmosphere, you tend to trap more heat than even the high levels of carbon dioxide, and the primitive Earth did have a high level of methane. Now, another macromolecule that is important to know is carbohydrate. And again, break apart the name. Carbo for carbon, hydrate for water. It has hydrogen and oxygen. And so these will have an equation where you'll have a carbon, and then you have 2 times as much hydrogen as oxygen. And so you can see that water molecule. It doesn't mean it's water molecule. It means that there's that 2 times as much hydrogen as oxygen. These are hydrophilic, which means hydro means water in this case, philic means loving, and so these can dissolve in water. If you think of sugar, you could put it in water, mix it, and it dissolves. Now, the most elementary carbohydrate that we talk about are called monosaccharides. Mono means one, saccharide is a single sugar, so a monosaccharide is one sugar. And there are two monosaccharides we tend to actually deal with. One of them is called a ribose. One thing you'll notice is this word ose is coming up here in the ribose and here in the hexose. That tells you it's a sugar. Now ribose is a 5-carbon sugar, which we'll talk about a bit later, and a hexose is a 6-carbon sugar. The main one that we talk about in biology is glucose, but you have a lot of them. So remember, a monosaccharide will be one saccharide, one sugar molecule, that's it. It can be a ribose or a hexose. Then we have something called a disaccharide. Anytime you see di in chemistry, it usually means 2. So disaccharides are 2 monosaccharides which are bound by dehydrating them. Dehydration means the same thing that you think, removing a water. So if you remove a hydroxide from one saccharide and a hydrogen from another, you pull water out and you glue them together, making a disaccharide. Now we can also have polysaccharides. Poly means many, saccharides again means sugar, so these are many sugars linked together due to dehydration. And you have a lot of different ones. One of them that is really important to know is starch. Starches are the polysaccharides found in plants, which we can break down. We actually have the enzymes to break these things down and use them as sugars. In animals, we can actually take the saccharides that we take in, usually in the form of, we'll take this polysaccharides and disaccharides, usually break them down to monosaccharides, absorb them, and we can take those and store them as glycogen. Glycogen is pretty much a big starch that animals can make to store sugars. If you look at the name, glyco will usually mean sugar. Gen means beginning. So glycogen is beginning of sugar. So what happens is you store the sugars as glycogen, and when your sugars are low, you make new sugars by breaking down the glycogen. Another 2 that are important to know is cellulose in particular, because it's made by plants as usually a structural polysaccharide. It is used in certain structures in plants, namely we talk about the cell wall, and these are not usually digestible by animals. So when you eat cellulose, we call it fiber, And it's important to maintain a normal, pretty much normal bowel movements. And they found that diets high in cellulose tend to actually decrease cancer rates, not just of GI tract, but of everything. Now, there are some animals who eat a lot of plants and use them for energy, but what usually ends up happening is they have a symbiotic relationship with bacteria in their gut and their stomach and intestines to actually help break apart the cellulose so they can use it. Without that, they wouldn't be able to use it. Sim means same, bio, life. So it's sharing life so both organisms can live. Not always, but in this case. And you do have chitin, which is used by fungus to make cell walls. It's also used by some animals as an outer shell. Now, in nutrition, we talk a lot about carbohydrates. Remember, the body cannot absorb the complex carbohydrates. We have to break them down to simple sugars or sometimes as disaccharides. Now, monosaccharides are the preferred energy source for almost all the body. There are actually some organs that if you do not have enough monosaccharides, they shut down. And when we're actually dealing with nutrition of carbohydrates, we usually are talking about calories as well. Calories are a measure of energy to produce heat. The measure is 1 calorie is the amount of energy needed to raise 1 gram of water from 25 degrees Celsius to 26. When you actually look at food packaging, they don't use a true calorie. They use what's called a kilocalorie, but they say, oh, it's a big calorie because they raise the calorie, the C in calorie as a capital, and so they say, oh yeah, it is a kilocalorie. A big calorie is a lot of times what they'll say. It is a kilocalorie, and the reason food companies don't want to use kilocalories is kilo scares people. When people hear kilo, they think it's big. So instead of using kilocalories, they lie to you and use calories with a big C. Now, there are 2 ways to break sugar. One of them is aerobically, and the other is anaerobically. Aerobic is here, anaerobic is here. Anytime you see aero like this, it means with air, and in metabolism, we're talking about with oxygen. Anaerobic, well, we have aerobic, and AN means without. So anaerobic would be without oxygen. And so an anaerobic breakdown of glucose, anaerobic respirations, we're not using oxygen. And I'll tell you a little bit about that in the next slide. Now, I'm going to start with the anaerobic breakdown of glucose because it is simpler and it gives you have to actually do it before you can go into the aerobic breakdown of glucose. Now over here I put glucose. This is the chemical structure for pyruvate, and I didn't put the chemical structure of lactic acid because we're just adding hydrogens. And so you have the glucose being broken down to 2 pyruvates. Remember, whenever you break something, you release energy. If you release energy, we call it an exo, outside, thermic heat reaction. So by breaking glucose into pyruvate and then adding hydrogens to it, we actually get exothermic reactions. We can take that exothermic reaction to run endothermic reactions, reactions that require energy to go into it. And the reactions that require energy to go in are going to build bonds. And so over here we have something called an ADP, which stands for adenosine diphosphate, and a phosphate, which I'll put an I here because it's an inorganic phosphate. A phosphate technically is PO4. And so the energy released by breaking sugar to lactic acid will take the ADP and the phosphate and make ATP. So you have an exothermic reaction paired with an endothermic reaction to make something. So you break down glucose to lactic acid, you end up making about 4 ATP. I'll talk more about that a little bit later. But I want to now move to the aerobic breakdown of glucose. Now, the first part of breaking glucose down so it can be used aerobically, you have to break glucose to the 2 pyruvates. That is an exothermic reaction, and then you can add a bunch of oxygen and take the pyruvate and break it down to carbon dioxide and water. A lot more reactions, a lot more breaking it down, so we get a lot more exothermic reactions. Exo means outside thermic, so we're going to release a lot of energy. And as we release a lot of energy, we take that energy, take our ADP and our phosphates, again, an organic phosphate, and we bind it to make ATPs. Remember, this is an endothermic reaction here. So an exothermic reaction will run an endothermic reaction. So when we break glucose to carbon dioxide and water, we get a lot more energy, and we can end up making anywhere between 24 to 40 ATPs in the complete aerobic breakdown of glucose. You hear me say aerobic respiration. The thing is, when we're dealing with breaking and down sugar for energy in cells, it is considered respiration. Now, the other molecule that I want to talk about are called lipids. Now, some of you have heard of words that have this in it, the lipo. If you think of liposuction, it means fat suction. So you're actually technically putting a tube jamming into someone and sucking the fat straight out of them. And so lipids are nonpolar molecules, which means that they are hydrophobic. Hydro means water, phobic means afraid of. And so if we take a lipid and we put it in water, it's going to separate. Lipids are also, something I should mention, lighter than water. And so what happens is, if something is lighter than water, it will float. It has a lower specific gravity, is what we'll call it in chemistry. In humans, we actually use lipids for energy a lot of times, and they're broken down through aerobic respiration. What ends up happening is, when you really want to break down pyruvate, you convert it to acetyl-CoA, and then you use oxygen to break it down. With fats, you take your lipids, you break them down to make acetyl-CoA, and then you use oxygen to break them down. Now, per gram, lipids have a lot more calorie than glucose. You're talking about 1 gram of lipids will produce 9 kilocalories of energy. What a lipid is it's pretty much a hydrocarbon, with a functional groups. Now, there are different functional groups. There's one called a carboxylic acid group, which is made up of a carbon and a hydroxyl group. And these will be bound to your hydrocarbons. You also have phosphate groups, which is a phosphorus with 4 oxygen, which will bind to fats. And it's actually kind of neat because phosphate is water soluble. It's hydrophilic. And so when you bind a phosphate to lipids, you end up having a molecule that has a part that's water soluble and a part that's fat soluble, or hydrophobic and hydrophilic. And then you also have glycerol group, which is a 3-carbon structure with 3 hydroxide groups attached to it. And these are going to act a certain way to bind what are called fatty acids, which I'll talk about in the next slide. Now, lipids. Basic thing you actually want to look at in lipids is the fatty acids. What they are is they're a carbon chain that can be saturated or unsaturated. If it's saturated, it means pretty much it has all the hydrogens it can store on that carbon chain. If it's unsaturated, it means that the carbons have a double bond somewhere. You can have in unsaturated, regular unsaturated, cis unsaturated, or trans unsaturated. Trans fatty acids tend to be synthetic, and there is a lot of issues with them. In fact, most industrialized countries have banned them. The US is one of the few that hasn't, and the reason most places have banned them is because they're linked to cancers, heart attacks, pretty much a lot of health issues. Unsaturated are usually better for you, but the trans are actually worse. Saturated are usually solid in room temperature, by the way. Now, besides the fatty acids, we have something called a triglyceride. Now, this doesn't mean 3 glycerides in this case. It has a glycerol group with 3 fatty acids, and so the glycerol group ends up binding 3 fatty acid tails. The phospholipid I mentioned earlier, it's a phosphate group with 2 fatty acid tails. And so it will actually form a molecule that's both water and fat soluble, called an amphipathic molecule, which I talked about in the past. And then the last lipids that we actually talk about are sterols, which, if you notice, sounds like steroids, and cholesterols do fall into this group, and they're a multi-carbon ring structure, which is a lipid. It's fat. And I will say, while some fats can be made by the human body, there are 2 essential fats that we cannot. One of them is called the alpha linoleic acid, and the other one is linoleic acid. And while you can make all the other fats, these 2 you need to have in your diet. These are also basically omega fatty acids, and so they're the good fats that people talk about in your diet. Now, we talked about how we can get fats from the body, from the diet, but we can also make fat by something called lipogenesis. Remember, LIP means fat, genesis means beginning. So what we can actually do is, let's say we took too much sugar in. We can take our fats, we can take our sugars, convert it to pyruvate, take the pyruvate, make it into acetyl-CoA, and then we can take the acetyl-CoA and make new fatty acids. And so that's why it's called lipogenesis. We're taking the pyruvates technically and making them into fatty acids. Remember, two of them are essential. The other ones we can actually make to a degree. Lipids in cells are actually really important because they make the outer membrane of our cells. The cell membrane of any type of organism, whether it's archaea, bacteria, or eukaryote, is mainly composed of phospholipids, that fatty acid tails with a hydrophilic phosphate. And as phospholipids are amphipathic, they will form two layers in water. One layer will have the phosphates, then the lipids, and the second layer will have the lipids and then the phosphates. So you end up making a pretty much a sphere here where you'd have water in here and over here. H2O. 2O. So you'd have water on both in here and here. The lipids would be on the middle, and then you have the, actually what I would do is bring this up. If we were actually looking at this structure here, we would have lipids here in the middle, two layers, and we'd have our phosphates, make them yellow for now, and make them a little bright red, over here. And we'd have phosphates here. So the water-soluble area would be facing both the outside here and the inside of the cell here, and the lipids would form a membrane. Now, this membrane is a lot of times stabilized by cholesterol molecules. It also contains specialized lipids which have sugars put in, which are called glyco, which means sugar, lipids. And they have proteins that are incorporated in there, which we'll talk about when we talk about proteins, like now. So we now have membranes that we can make in our cells, made out of phospholipid bilayer. Now the next carbon structure I want to talk about is amino acids. Now, amino means it has a amine group, which is a functional group with nitrogen and hydrogen on it. And as it's an acid, it will have a carboxylic acid group, which we've talked about before. Carbon with a hydroxyl group associated with it. And the amine group has the, pretty much similar to ammonia, nitrogen bound to two hydrogens. And this actually gives a structure which is really neat because it has these two things, and a lot of times it'll have a third structure which is carbon-based, but it can have sulfur and a lot of other things. Now the neat thing about amino acids is they can make certain special bonds called peptide bonds. Now what happens is you take the one end of the carboxylic acid end, you take a hydrogen here, you pull it off, making water, and then you end up actually having a peptide bond, and the molecule will have two amino acids, which we call a dipeptide. So again, what it's going to happen is the hydroxyl group from the carboxyl group combine to a hydrogen from the amine group, leading to a dehydration reaction. And so the two amino acids become a dipeptide. When you have multiple amino acids glued like that, we call it a peptide, a polypeptide. And when we have a lot of amino acids or a lot of polypeptides clumped together with a bunch of different structures, we call it a protein. Now the interesting thing about the human body is we can use amino acids for energy, but we cannot absorb proteins directly. For us to absorb a protein, we have to break it down to amino acids or dipeptides, and that's what we can absorb. Interesting thing is each amino acid, when used for energy, can end up giving you about 4 kilocalories per gram. Now, I will tell you that there are many amino acids, but there's only 10 essential. And you can get the 10 essential amino acids from a lot of different things. So there are vegetarians out there who can tell you how many times someone told them that they can't get all the proteins that they need. Well, you don't need the proteins, you need the amino acids. And as long as you're mixing your foods, as long as you're looking at where your 10 essential amino acids are coming from, you can be as strict as a vegetarian or vegan as you want. Doesn't really matter because you just need to get the essential amino acids. Now, I am just going to mention a couple specialized cellular proteins, and one of them is enzymes. Enzymes are proteins that speed up a reaction, and what that's called, anything that speeds up a reaction but doesn't get used up itself, is called a catalyst. So enzymes are protein catalysts, and whenever you see ASC at the end of a word, it usually means that they're protein catalysts or enzymes. You also have specialized proteins that give your cells, any cell technically, a shape, and they are called cytoskeleton. Anytime you see cyto in a word, it usually means cell. So cytoskeleton protein is a protein that makes a skeleton of a cell. Remember, in biology, the words are not just words, they're sentences. Now, there are some proteins that are going to be associated and incorporated into the cell membrane. Some of these are called peripheral proteins, which they're technically just on the periphery of the cell membrane. And some of them are called transmembrane proteins. Again, remember, trans means across. So transmembrane proteins will be proteins that go across the cell membrane. A lot of these will form pores through the cell membranes, which we'll talk about in a little bit. Now, the last macromolecule that I am talking about today are called nucleic acids. Nucleic acids are composed of a sugar, a phosphate, and a nucleotide base. Nucleotide base, I will talk a little bit about later. It's actually just a structure at the end which can form hydrogen bonds. A nucleotide and has nitrogen, by the way. Our sugar that we use is ribose. Now remember, os means sugar, ribose is a 5-carbon sugar. And there are 2 nucleic acids that we deal with. One of them is called ribose nucleic acid, tells you the sugar is ribose and it's a nucleic acid. And there are 4 variants based on the nucleoid base that's associated with it. You have adenine, cytosine, uracil, and I put it bold for a reason, and guanine. Now, if you look at this, adenine forms 3 hydrogen bonds, cysteine forms 2, uracil forms 2, guanine forms 3. If you actually do have a double-strand RNA molecule, which happens when you're making certain things, the adenine and the guanine are, and the uracil are going to bind. The cysteine and the somewhere that doesn't make sense. I put these wrong. The cysteines are going to bind together. The uracils have 2. Adenine, this should be 2 here. Sorry, and the cysteine should be 3 hydrogen bonds. Again, this in the DNA, this should be 2 hydrogen bonds, and cysteine should be 3. I must have hit the wrong number. And so you are going to form different bonds. Usually when they're bound, the adenine will go with the uracil, the cysteine will go with the guanine. In DNA, which is going to be double-stranded, adenine go with thymine, and cysteine will go to guanine. If you notice, the thymine is replaced by uracil in RNA. Now, let's go a little bit more into ribose nucleic acids. These are used as a plan to make proteins directly by specialized enzymes. The specialized enzymes are called ribosomes. And one of the things I want you to pay attention with ribosomes is you see this word here, ribose, which tells you it's associated with the ribose sugar. So ribosomes are going to take RNA and they're going to make a protein based on what the RNA molecule tells it to make. RNA pretty much is read 3 bases at a time, and every 3 base encodes for 1 amino acids. That process is called translation. So you're going to translate an RNA molecule and make a protein out of it. 3 nucleotide codes encode for 1 amino acid, which are glued together in peptide bonds, which we talked about. In the deoxyribose nucleic acids, this is actually the genetic material that we inherit. All life, in this planet at least, inherits DNA as its genetic code. Your DNA has all the proteins that you need for life. And they are bound complementary. So the T bind to the A, G bind to the C. When you're making proteins, you do it in a process called transcription. So what you're going to do is you're going to take the DNA here and you're going to bind it to RNA. Now the A is going to bind to U, G is going to bind to C, and the T is going to bind to A. Remember, no T's, you have U instead. The G's and C's are always going to bind, so the C will bind to a G too. Now, how this works. As DNA is a double-stranded structure, it's held together by the hydrogen bonds. And so when we want to make it, we have to melt it, or we have to, what I mean is we have to break the hydrogen bonds to allow things to go in there. It's going to bind to the complementary pair. So the C will bind to the G and so on. When we're making proteins, the same thing happens, except we make RNA from the DNA first, and that's actually what you're going to use to make your proteins. Making RNA from a DNA template, again, A's will bind to U's, T's will bind to A's, G's will bind to C's, C's will bind to G's. And the interesting thing is the enzyme, the protein catalyst that's going to glue the bases together is called an RNA polymerase. The reason I mention this is RNA. It tells you you're going to be using RNA. ACE tells you it's an enzyme, and it's going to make a polymer, which means it's going to put a lot of these things together, a lot of these bases together. So poly, many units. Again, when we're making DNA, we open up the DNA so the double bonds break, and the complementary bases will actually bind to the original DNA strands. Now you notice I put strands with an S here. The reason is you're going to use both DNA strands to make copies. The DNA strands are then going to be glued together by something called a DNA polymerase. Again, ACE is an enzyme, and it's going to make a polymer, so it's a polymerase. Now we can actually go back to our basic cell membrane. It's usually referred to as a fluid mosaic model, and if you think of mosaic, it has multiple parts involved in it. So you're going to have the phospholipids, phosphate heads on both sides. I'm not going to draw a lot of them. You're going to have lipid tails coming 1, 2 for each one, 1, 2, and this will actually continue all the way around and making a hollow sphere filled with water, actually, technically. And then you're going to incorporate stuff into it. So we're going to actually put some proteins in here, which go all the way through. So there would be a transmembrane protein. We might have a protein that just sits on the outside, linked in, which is a peripheral protein. We'd put some cholesterol molecules in here, those ring structures in here to help stabilize everything. And we'd have something called glycolipids in there, fats with sugars attached to it. So we'd have fatty acids, and then we'd have sugar molecules. I'll make them orange so we can see them a little better, coming out here. And as you have a fat sandwich, pretty much, water-soluble bread to fatty acid tails, we end up having something called a semi-permeable membrane. As the inside is hydrophobic or lipophobic, depending on how, it tends to keep large hydrophobic molecules out. It also will allow some fat-soluble things to be able to go through, as long as it's being carried by something. And to some degree, water will be able to move across, and that will become important later. Now, what I want to talk to you about is also something that I've mentioned before, and that's diffusion. Again, remember, diffusion is a passive movement of molecules from an area of high concentration to low. I gave you the idea of scent. If you smell something, it goes from high concentration to low. Another example you might look at is, think of food that went bad that's in a sealed container. If it's in a sealed container, it won't be able to get out. But the minute you puncture a hole in it to see what's inside, that can come out. And so, as you know, if something has a way to go through, it can, and it will. In the cells, we have another way we can actually, another thing we can do. We can have the diffusion, but we can actually have something called facilitative diffusion, which if a molecule is too big to go through the cell membrane, we can put a protein pore in it that will open and close, that will allow things to come in and out. The other one that's important for you to know is osmosis. Osmosis is a net movement of water across a semi-permeable membrane caused by the solutes, or dissolved molecules, different on the opposing sides. So if you have more sodium or you have more molecules on one side of a membrane than the other, it can actually cause a pull. The more solvents, the solutes, can actually cause a pull on water. Now, we do have something that's very important for you to know, and I would tell you to write it down and understand it, and that's your osmolarity. That's the number of solutes per liter. Now, one of the things with osmolarity is moles can have different osmolarities. For instance, glucose has a osmolarity of 1, meaning that when you put it in water, it stays 1 mole of glucose. If we have sodium chloride, for instance, and we put 1 mole of sodium chloride in water, sodium chloride breaks apart. So sodium chloride will actually have 2 molecules dissolved per mole, and so you're going to have an osmolarity of 2. So it has to deal with, the osmolarity will change depending on how the particles are dissolved in water, how they dissolve. Now, as we have now a cell membrane and different things, we can talk about osmosis. We can talk about diffusion and how there's different types of diffusions. You actually can have facilitated diffusion or diffusion just through the membrane. Small molecules like carbon dioxide, for instance, can cross through the cell membrane. Sugars won't be able to. Sodium and chloride will not necessarily be able to diffuse. And so sometimes you have passive diffusion channels which allow co-transport, facilitated diffusion. And the facilitated diffusion, because of the protein, can end up coming in together, which are co-transports as well. What co-transport is, as you have different concentrations of something on the outside and inside, two things can actually be brought in together. We also can bring things in through active transport. Based on the word active, it's shown that you require energy. You actively have to pump something to go against the concentration. So if you have more of something inside a cell than outside and you want to bring it in, sometimes you could bring it in through a co-transport facilitated diffusion, which would be passive, or we could do an active transport where we're breaking ATP. So we take ATP and break it to ADP and phosphate to get it in. Sometimes the active transport can also be a co-transport, which I'll tell you a little bit later. So co-transports can either be facilitative or active. The way that it can be facilitative is you can have two molecules that are bound and are brought in together because one has a much higher amount of particles outside a cell than inside. So an example of that is there's a special protein called a sodium glucose cotransport, which takes sodium and binds the reaction with glucose, and as sodium usually is higher outside the cell than inside, when you have sodium and glucose bound to a protein, the channel will open and get the glucose to follow the sodium in. So it's a cotransport which is passive, but it uses the concentration difference. Some cotransports require ATP to move two different molecules. The one I love to talk about is a protein called sodium potassium ATPase. It takes 3 sodium, you can see Na here, there's a sodium, and 2 potassium, and uses ATP to move them across the membrane, causing different concentrations of sodium inside the cell than outside, as well as different potassium concentrations inside and outside. So if you're pumping 3 potassium out and 2 potassium in, eventually you're going to have a lot higher sodium concentration outside the cell than inside, and as you're pumping 2 potassium inside the cell, you're going to end up having more potassium inside the cell than outside. Now, why this is important and what it means for the cell membrane is, first of all, you're going to have a difference in concentration of these ions. And so you can use, like I mentioned earlier, the difference in sodium outside and inside to pull things in by opening sodium channels and allowing sodium to move in with something else. The other important thing is we're pumping 3 sodium and 2 potassium. And so besides the concentration difference, we're also having more positives leaving than positives going out. So we end up having a net negative charge difference across the membrane. So because of this, as we're pumping 3 positives out and 2 positives in, the membrane will end up having a charge when it's resting. When it's resting, it doesn't mean it's not doing anything. It means it's getting ready to do something. And so we call that charge the resting, the cell is not doing anything except for setting itself up, membrane, it's on the membrane, potential. So it's a negative charge on the membrane when the cell is at rest. This also, again, leads more sodium inside the cell than outside the cell. I'm sorry, more potassium inside the cell than outside the cell, more sodium outside the cell than inside. Now, I want you to think of, we have a cell that is at rest, and the sodium Na, potassium K, ATPase, enzyme that breaks ATP, is active. So we've been pumping sodium out, potassium in. If we started with the same concentration of sodium inside and outside the cell, what would happen to the amount of sodium inside the cell as we're running this? Think about it. If you're constantly pumping sodium out, it's going to end up causing you to have lower sodium inside the cell, so the concentration of sodium outside will be higher. If we look at the potassium, if we've constantly been pumping potassium inside the cell, the concentration of potassium inside the cell will become higher than the potassium outside the cell. And so what would happen if we opened up a sodium channel on the cell membrane that moves on diffusion if we have more sodium inside the cell than outside? The answer would be, based on diffusion, molecules move from higher concentration to low, so the sodium would actually go from the inside of the cell and go outside the cell. If we open up the potassium channels, as we've been pumping, if we've been pumping sodium out of the cell, when we open up the sodium channels, we will actually get sodium to flood into the cell because of diffusion. If we open up a potassium channel and we've been pumping potassium into the cell, then potassium will leave the cell when we open a passive channel. Always remember what the passive force that drives this movement of potassium and calcium in or out of the cell. If we have more concentration inside the cell, like in potassium, the potassium will flow out based on the passive movement of molecules from high to low concentration would be diffusion. The passive movement of sodium, because of high concentrations outside to the low concentration inside, so sodium going into the cell, it would also be diffusion. And the reason we can do that is because at rest, we've been pumping sodium out and potassium in.